So in this past slide, we talked about the bile acids. So I'm going to talk about the implications and importance of bile acids by going through the biliary system and how the bile affects the gut and the liver. So just a little back to human physiology and what the purpose that bile acids serve. Bile acids, they're made in the liver from cholesterol. They're conjugated to amino acids like glycine and taurine. And once they get conjugated, they're called primary bile acids or conjugated bile acids. These bile acids, they're concentrated in the gallbladder. And as food moves into the first part of the small intestine, the duodenum, it triggers the release of cholecystokinin, and that causes the gallbladder to release these primary bile acids into the duodenum. And the main purpose there is to facilitate the digestion of fats. So fatty meal comes along, and this is why when someone has pancreatitis or cholecystitis or gallbladder removal, they often have a hard time breaking down fats because we need the cholecystokinin and the bile to break down fats. So the bile moves into the digestive tract, helps break down the fats, helps absorb the fats. And not only that, but also cholesterol and other fat-soluble vitamins. So your A, D, E, and K helps with the absorption of those. What's interesting about the bile acids is about 95% as they move through the digestive tract are reabsorbed and transported back to the liver through the enterohepatic circulation in the portal vein. So we can see here, we've got our green gallbladder releasing the bile. Bile acids move into the digestive tract. 95% of them go back to the liver and can be reused to make bile and bile acids again. 5% goes to the stool. And in the stool, those primary bile acids do have a role. They turn into secondary bile acids, and they could affect the microbiome and actually have a slight antibiotic effect on the biome to prevent potentially pathogenic bacteria from creeping up in abundance. So the biliary tract, the liver transports the bile salts and antimicrobial molecules like IgA, angiogenin to the intestinal lumen. This helps maintain what they call a eubiosis, a healthy, happy, good microbiome. It prevents overgrowth of the bacteria, so it kind of keeps SIBO or small intestinal bacteria in check. Not only the biosalts help with the uptake of fat and keeping a healthy microbiome, they also act as important signaling molecules. There's two receptors. One is the FXR receptor, and the other is TGR5. We'll talk about those. I talk about those a little bit more in the PowerPoint, but they really help to modulate. They target these nuclear receptors, and they modulate hepatic bile acid synthesis. So it's a feedback loop. The bile acids target these receptors, and then they prevent the liver from overproducing bile acids, because the accumulation of bile acid in the liver can cause liver inflammation and promote oxidative stress. And that oxidative stress and inflammation can lead to hepatotoxicity and further potentiate liver disease like biliary fibrosis and cirrhosis. So the feedback loop of bile acids having that negative feedback reduces the risk for liver disease. We talk a lot about the gut microbiome, even the skin microbiome, the vaginal microbiome, but there's a bile microbiome. And the existence of the bile microbiome is starting to come into prominence. People are learning more and more about this. And it was shown that healthy people, for the first time, were shown to have a bile microbiome when samples were taken and on resections of the gallbladder. So the surgery was performed, gallbladder was resected, and they started to find that there is a true microbiome there. It does have a similar composition to the gut. So it has a lot of Firmicutes, Bacteroidetes, Actinobacteria, and Protobacteria. Those are the dominant species, and those are similar to the dominant species that we see in the gut. However, they noticed in patients with one form of liver gallbladder disease, the primary sclerosing cholangitis, these patients, there was decreased alpha diversity and expansion of pathobionts in the bile compared to healthy individuals. So what does this mean? A couple interesting terms here: alpha diversity and expansion of pathobionts. So alpha diversity is the overall diversity of the various strains of bacteria within an ecosystem. So they say that in someone with a healthy gut or someone with a healthy gut microbiome, they have a strong alpha diversity. They have a wide variety of healthy bacteria in the gut. Here in these primary sclerosing cholangitis patients, that diversity of various types of bacteria was diminished. Also, expansion of pathobionts. That means there's these biological forms, bacteria that can elicit a pathology. They're named pathobionts. So microbiologists, they define this term as any potentially pathological organism which under normal circumstances lives as a non-harming, it lives in symbiosis with the host. However, when they expand and creep up in size or abundance, they can cause pathology. Not only was the taxonomic composition, so not only was the diversity altered, but the function of the microbiome, the bile microbiome, can be altered when the bile microbiome, when the bile microbiome is out of balance, the function of the biome itself is also inappropriate. And this leads to an increase in pro-inflammatory bacterial pathways, and this further promotes inflammation and liver disease within the liver. This just is more of the same from the last slide, that altered bile microbiome can be correlated to liver disease and can lead to complications. So we can see that cholestatic liver disease exhibits a variation in bile acids, the size and the composition of them. Dysbiosis of the bile microbiome is also seen in cholestatic liver disease. And these changes have been shown in research to play a role in the pathogenesis and progression to more chronic cholestatic disease states and progressed liver disease states. So it's interesting, not only does the gut microbiome have an important role, but the gallbladder and bile microbiome is playing a role on the liver disease. So that bidirectional crosstalk, it's not just what happens in the gut goes through the portal vein to the liver, but what's happening in the biliary tract with bile is backing up and affecting the liver as well. When these bile acids return to the liver, 95% of them, they inhibit cholesterol and further bile acid biosynthesis. So in a previous lecture in the hepatology modules, it was discussed how the importance of fiber, dietary fiber binds up cholesterol and bile acids, sequesters it, and pulls it out when someone has an elimination, when they have a bowel movement. So here, without that fiber, these bile acids, they're returning to the liver. Yes, they can be reused to make bile. They also have that negative feedback that inhibit cholesterol. Small quantities reach the colon. Most of the small quantities of primary bile acids that reach the colon in large intestine are converted to the secondary bile acids. And these bile acids in the colon eliminate bilirubin, excess bilirubin from the body and the stool. That's what gives the stool its brown color. But importantly, the secondary bile acids have that strong antimicrobial activity and cytotoxicity. So this helps modulate the microbiome and protects against ascending infections going back up in the biliary tract, biliary tree. How do these bile acids affect the immune system? Well, cells of the innate immune system, macrophages, dendritic cells, and natural killer cells, many main cells of the immune system, they have receptors for both primary and secondary bile acids. So I mentioned before the FXR and the TGR5 receptors. This stands for farnesoid X receptor, that's the FRX, and the G protein coupled receptor. This is actually a Takeda G protein receptor. They are receptors for the bile acids, and when bile acids bind to these receptors, they actually promote an anti-inflammatory response. There's a shift to downregulate the cytokines, the inflammatory cytokines. So we can see here, these initials LCA and DCA, these are various bile acids. As they come and they encounter immune cells, they bind to these receptors, and we can see interleukin 10 goes up, which is anti-inflammatory. Tumor growth factor beta goes up, also anti-inflammatory, but we can see there's a response. Interferon gamma, a main pro-inflammatory molecule, is decreased. TNF alpha, interleukin 6, interleukin 1b, all pro-inflammatory cytokines, they're reduced as bile targets these receptors, and that reduction in these cytokines reduces inflammation in the liver. So the bile acids have a strong play on the effect of inflammation that's happening in the liver and liver disease patients. Just some notes on patients with non-alcoholic fatty liver disease and other liver diseases. It's been shown in the research that the amounts of bile acids, especially secondary bile acids, increases in those with liver disease. Also in metabolic syndrome, bile acid metabolism is altered, and it's shown in correlation with hepatic steatosis. Changes in the composition of bile acids can allow unusual bacteria in the intestinal microbiota to proliferate and ascend into the biliary tree, creating inflammation in the biliary tree and a dysbiosis in that biliary tree. And there's evidence that 1, microbial agents, 2, changes in the microbiota, and 3, altered metabolite profile of bile acids can lead to primary biliary cirrhosis and primary sclerosing cholangitis. So it shows that these microbial agents can promote liver disease. One more slide on bile acids. Dietary fat consumption. So eating overconsumption of dietary fats promotes the production of the bile acid taurocholic acid. This is a particular bile acid. And TCA production can increase a particular bacteria called Bilophila wadsworthia. This is a sulfite reducing bacteria that promotes intestinal permeability and inflammation. So what this slide is saying is those that overconsume fats are likely to produce this particular bile acid, the taurocholic acid, TCA. TCA is likely to increase the Bilophila wadsworthia, which promotes intestinal permeability, and that intestinal permeability can lead to inflammation in the system. The TGR5 receptor being overstimulated by the bile acid, it's not properly activated, and that causes white adipose tissue browning to be inhibited, decrease in secretion of glucagon-like peptides, so the GLP-1, and decreased secretion of insulin. So all of these are promoting obesity and insulin resistance, all from bile acids.